Yeah. Hi, everyone. So our research is about using topological data analysis to analyze neurodegenerative disease and genomics. So our team, it consists of Krishnaveni, Sathya, Deniz, Aparna, me, Ria, Simone, and Kevin, and our principal investigator is Ms. Sahar Jahanikia.
Yeah, so our project was basically using TDA to analyze neurodegenerative diseases. So neurodegenerative diseases, they often have underlying genetic causes. They're hard to exactly pinpoint and analyze. But TDA, which is a topological data analysis, it uses persistent homology, which is a branch of topology that studies mostly like underlying space, and then computational geometry to provide insight into the underlying structure, shape and structure of data. And so applications to cancer research and disease analysis have shown the value of using TDA to identify significant patterns in genetic markers. And so we plan to use TDA to analyze genomic data in order to better understand how these neurodegenerative diseases occur. And we're using TDA algorithms such as Kepler Mapper, and we're analyzing the structure of these specific areas of the genome that are associated with these diseases. And hopefully through our work, we can help better improve diagnosis and then find better respective treatments.
Okay, so to start, we're going to start with genome. So genome just contains all the genetic information of an organism. Genome sequencing is a laboratory procedure that determines the order of nucleotide bases in a genome. And then gene expression is defined as the process by which instructions in DNA are converted into a functional product. So that can mean a protein, which then causes the gene expression.
So the neurodegenerative diseases that you see here are some of the ones that we will be analyzing. They consist of Huntington's, Alzheimer's, Parkinson's, Lewy body dementia, and multiple sclerosis, among others as well. And we have already identified some of the genes that are associated with these diseases, and they will be used in our analysis.
So now I'm going to be talking about the principles of topology. So one of the first principles is called coordinate invariance. And this basically is the principle that our data, or shape of data in this case, doesn't depend on the coordinate choice we use. So we don't have to take into account position, and the shape of our data stays the same. The second principle is called deformation invariance. And this is the principle that stretching or compressing a shape in a plane doesn't change its topological structure. So to decode that a little bit, basically the shape of our data doesn't change by how we alter or deform that data in itself. So our structure remains intact. And the final principle of topology is compressed representations. This basically says that some details can be sacrificed while maintaining the shape's integrity. So our shape is preserved while we can compress down the data into a more usable form while keeping most of the more important details.
There are two different aspects of TDA that we will be using in our analysis, which are dimensionality reduction and Kepler Mapper. Dimensionality reduction is the process of reducing noise in high-dimensional data by decreasing the number of variables. Data with an excess amount of noise can cause overfitting in machine learning, inaccurate data analysis, and increase in time complexity. Luckily, dimensionality reduction provides a solution to all of these. There are many methods of dimensionality reduction, but the one we will likely be using is uniform manifold approximation and projection, or UMAP. So Kepler Mapper is Python's implementation of TDA's mapper algorithm. Basically, mapper is a function that builds upon any dimensionality reduction algorithm like UMAP and produces a low-dimensional simplicial complex to represent the data. And you can think of a simplicial complex as just a set of points. Mapper is really useful because it can be used to identify interesting topological structures in data. How the mapper algorithm works is, first, it divides the data into overlapping intervals, and then all data points within an interval are collapsed into a single node, and all nodes with overlapping intervals are then connected with an edge. And an example is over here, and you can see that the final structure preserves the overall global structure, although it does leave out some information.
I'll be talking about the methodology. So our research aims to identify genetic indications in neurodegenerative diseases, and as well as discovering and for drug discovery. So we first started off by researching various neurodegenerative diseases, and we also researched some genome sequencing and basic genome knowledge. And we also researched the basics of TDA. In the near future, we plan to apply TDA to our own data, but for now, we're applying it to sample data for practice. And we also are using dimensionality reduction and mapper algorithm. With that, we'll analyze the shape and the underlying structure of large genomic data sets and identify patterns. The analysis will be done in Python using scikit-TDA. However, we're also finding new libraries and programming languages such as R. Our eventual goal is to create a map like the one shown below to genes linked to the neurodegenerative diseases. We're hoping that our research will have some significant results and implications in medical sciences.
So far, we've researched neurodegenerative diseases such as Alzheimer's and Parkinson's, and we researched the basics of TDA. And now currently we're researching libraries to apply TDA, and we're gonna perform TDA on sample data. And then afterwards, we'll clean up the data we have from the genes responsible for the neurodegenerative diseases we're using. And we use Kepler Mapper, which is a Python library to analyze our data using TDA. And then we'll probably draw conclusions from the maps we made and then identify those implications.
So in terms of the technology we're using, we're mainly using Python to analyze our data, and we're running Python on Jupyter Notebook and Anaconda. We're also using R for some data cleaning and just comparing it with Python to see which one is more efficient and which one will suit our project better. In terms of the actual TDA and Kepler Mapper, we're using scikit-TDA and scikit-TDA's inbuilt mapper function. Those are the two logos on the right. And essentially that creates that interactive map that Deniz and Kevin were talking about, and it allows you to see sort of the structure of the data split into the nodes. So yeah, for our acknowledgements, we would like to thank our advisor, Sahar Jahanikia, for her extensive support and guidance. She continually mentored us throughout this summer. We're a newer group, but we were able to accomplish so much with her help. And then we would also like to extend a huge thanks to ASDRP for giving us the resources to pursue this project. And all our citations can be found at the QR code below if you want to take a look. Thank you.